Lysosomes are an animal cell's digestive organelles. A typical lysosome contains at least 50 different hydrolytic enzymes produced in the rough ER and targeted to these organelles. Taken together, lysosomal enzymes can hydrolyze virtually every type of biological macromolecule. The enzymes of a lysosome share an important property. All have their optimal activity at an acid pH and thus are acid hydrolases. The pH optimum of these enzymes is suited to the low pH of the lysosomal compartment, which is approximately 4.6. The high internal proton concentration is maintained by a proton pump present in the organelle's boundary membrane. Lysosomal membranes also contain a variety of highly glycosylated integral proteins whose carbohydrate chains are thought to form a protective lining that shields the membrane from attack by the enclosed enzymes. Although lysosomes house a predictable collection of enzymes, their appearance in electron micrographs is neither distinctive nor uniform. The presence within a cell of what is, in essence, a bag of destructive enzymes suggests a number of possible functions. The best studied role of lysosomes is the breakdown of materials brought into the cell from the extracellular environment. Many single-celled organisms ingest food particles, which are then enzymatically disassembled in a lysosome. The resulting nutrients pass through the lysosomal membrane into the cytosol. In mammals, phagocytic cells, such as macrophages and neutrophils, function as scavengers that ingest debris and potentially dangerous microorganisms. Ingested bacteria are generally inactivated by the low pH of the lysosome and then digested enzymatically. Peptides produced by this digestive process are posted on the cell surface, where they alert the immune system to the presence of a foreign agent. Lysosomes also play a key role in organelle turnover, that is, the regulated destruction of the cell's own organelles and their replacement. During this process, which is called autophagy, an organelle is surrounded by a double membrane structure to produce a double membrane sequestering vesicle called an autophagosome. Once formed, the outer membrane of the autophagosome fuses with a lysosome, generating a structure called an autolysosome, in which both the inner membrane of the autophagosome and the enclosed contents are degraded. The products of these degradative reactions are made available to the cell. It is estimated that 1 to 1.5% of the proteins within a healthy liver cell are degraded via autophagy per hour as part of a normal process of cellular renovation. Enclosure within autophagosomal vesicles is thought to be relatively selective rather than simply a random process. Autophagy probably evolved in early eukaryotic organisms as a response to nutrient deprivation. If a population of cells is placed under starvation conditions, a marked increase in autophagy is observed. Under these conditions, cells acquire energy to maintain their life by cannibalizing their own organelles. The same autophagic process is induced in the cells of a mammalian embryo before it is implanted in the uterus and thus before it can take advantage of nutrients delivered via the mother's bloodstream. Our understanding of the importance of autophagy in animals has coincided with the discovery and analysis of a host of genes that are required for various steps in the autophagic pathway. Deletion of certain of these autophagy genes can have serious consequences for embryonic development or adult physiology of model organisms. It has been shown that autophagy helps protect an organism against intracellular threats ranging from abnormal protein aggregates to invading bacteria and parasites. Autophagy has also been implicated in the prevention of neurodegeneration. If autophagy is blocked in a particular portion of the brain of a laboratory animal, that region of the nervous system experiences massive loss of nerve cells. And these findings reveal the importance of autophagy in protecting brain cells from the continuous damage to proteins and organelles that is experienced by these long-lived cells. Autophagy may even play a role in the prevention of certain types of cancers and slowing the body's aging process. The role of lysosomes in autophagy is summarized as follows. Once the digestive process in the autolysosome has been completed, the organelle is termed a residual body. Depending on the type of cell, the contents of the residual body may be eliminated from the cell by exocytosis, or they may be retained within the cytoplasm indefinitely as a lipofuscin granule. Lipofuscin granules increase in number as an individual becomes older. Accumulation is particularly evident in long-lived cells such as neurons, where these granules are considered a major characteristic of the aging process.